Okay, so here we're going to have an introduction to bacteria. And so the first thing to understand about bacteria is how they are different from us, because many of their differences are the target for our current antibiotics and developing antibiotics. So when you think about a bacterium, they are, of course, prokaryotes. They are single-celled. They have no um, organelles inside, no membrane-bound organelles. So they're very different than eukaryotes from that perspective. So eukaryotes are going to have mitochondria, which is a remnant of a gram-negative bacterium. And this is going to be the site of cellular respiration, whereas your prokaryotes, your bacteria, do not have a mitochondria, but they do cellular respiration. They just use their cell membrane to do cellular respiration because that's the only membrane that they have. So cellular respiration is where you generate a lot of ATP. So bacteria are able to generate a lot of ATP with cellular respiration by using that cell membrane. Now, prokaryotes also are going to have genetic material that's a little bit different than eukaryotes. So prokaryote genetics is going to include the chromosome, which is typically here the DNA, of course, plus any plasmid, so extra plasmids. And these plasmids typically are going to have virulence factors. And they can have antibiotic resistance. They can encode for a capsule, for example. So these extra plasmids are really going to help bacteria gain properties that enable them to survive better. Now, the other thing that prokaryotes have are, of course, flagellum. So these will help them to move. Not all bacteria have them, but some do have them. And then the bacterium are also going to have a peptidoglycan layer on their cell wall. And this is unique. So this is a target of our beta-lactam antibiotics, our penicillins, and the like. They also have a different size ribosome. So they have a 70S, whereas we have an 80S. So that is another potential target for an antibiotic. And so just these simple differences between prokaryotes and eukaryotes have led to the use of our current arsenal of antibiotics to target these pathogens. So the next thing to talk about is just the size of bacteria as well as their shape. And so typically we think of size as whether we can see it, and so we cannot see bacterium using our human eyes. We will need to use microscopes to see bacterium. So our fungi, our protozoas are at that edge of being able to see, and then our bacterium will be required for our light microscopes. And then, of course, our viruses are smaller. Some of the smaller bacterium are the rickettsia, chlamydia, and mycoplasma, and they're sort of right at that threshold of being able to view under light microscope or requiring an electron microscope to see them. Now, the other thing is that we have different sizes and shapes. So over here we have our cocci, and these are just round. And you can see that we have clusters, and we have strands. We can have diplo, which is just the two, and they can be diplo in various ways. Staph likes to cluster, and it's a cocci, and our strep likes to be in a strand, and it's a cocci as well. And then, of course, we have our rods or bacilli. So these are rod shapes. So these are usually elongated. They're not the nice little rounds. You can have sort of these coccobacilli. which are not quite round, but they're also not really the long, nice bacilli. They have rounded edges. You also have our spirochetes. And then you have a comma-shaped, which is cholera. Spindles. So again, you can have many different shapes associated with bacterium. Now the next thing to cover is our gram stain, because a gram stain is really a way to see these shapes. But keep in mind that the gram stain, you have limited information you can get from the stain. The gram stain is considered a differential. It is able to tell the difference between gram-positive and gram-negative. And basically for a gram stain, you start with a smear on a slide, and then you are going to stain with a purple color, which is called crystal violet. And then the crystal violet will stain the cell wall of the bacterium. So here's our bacterium. And they're going to have these peptidoglycan layers. And so the crystal violet, which is purple, will get into the peptidoglycan layers. And then you're going to add an iodine. Iodine is very large and it's a mordant. So iodine will come in, I'll make it a different color. Iodine will come in as a mordant and get stuck in a thick peptidoglycan layer. In a gram-negative, you have a very thin peptidoglycan layer, and it's in between. And so the purple doesn't really get stuck. The iodine doesn't really stick. And so when you do the next step, which is your alcohol destaining, basically for a gram-negative, so here's a gram-negative, here's a gram-positive, the gram-negative will lose the purple. And then the gram-positive will retain the purple. And so after you do a counter stain with pink, and then the pink will stain the gram-negative, and then the purple is still remaining the gram-positive. So when you look at this picture, they look like purple, and if you had a higher magnification, these might be cocci. They could be a coccobacillus as well. You can't really tell at this magnification. But what can a gram stain tell you? A gram stain tells you if it's gram-positive or negative, the shape of the bacteria, and some other properties, whether it's clusters, a diplococcus, etc. A gram stain cannot tell you the type of bacteria. You cannot look at a gram stain and see clusters and say, oh, it must be Staph aureus. You need to do other tests in order to determine if it's Staph aureus. You cannot look at a gram stain and say that, oh, they're comma-shaped, they must be cholera. You still have to do other. You can have an idea, perhaps, of which way to test for next, but it is not a diagnostic test. Gram stains are not used for diagnosis. All right, so let's look at the different gram-positive versus gram-negative. So here's a nice picture of it. So again, you have your peptidoglycan, which we'll talk about in just a moment. You have other acids on the outside, teichoic acid, lipoteichoic acid. And so your gram-positive bacteria are going to have one cytoplasmic membrane, and then all of this exterior properties are outside of that membrane, so they're bound to that membrane. So gram-positive bacteria are said to have a thick peptidoglycan layer. Okay, but it's important they only have one membrane. And then our gram-negative bacteria looks a little different. Our gram-negative bacteria have an outer membrane, and they have that same cytoplasmic membrane that you saw in the gram-positive. Now, what this outer membrane does is it adds a little bit more complexity to the gram-negative bacterium. And as I mentioned in a previous lecture, that outer membrane really allows these bacteria to survive. Our stomach acids get into the small intestine and cause a colonization and infection. Some of our famous gram-negative bacteria, of course, are E. coli, Salmonella, Campylobacter, Shigella, cholera, etc. Okay, the famous gut bacteria. And so a few things to point out here is there are some different structures associated with gram-negative bacteria. You don't see teichoic acid and lipoteichoic acid, but you do see LPS, and this is an endotoxin. Now, why this is called an endotoxin is an endotoxin is part of the bacteria that can be toxic to us. So it's not like a toxin like cholera toxin where their bacteria is producing it, releasing it. Cholera toxin can potentially affect the bacterium negatively as well. LPS never affects the bacterium negatively. It is part of the bacterium. It's part of their surface proteins. It is part of their being, and so therefore it is referred to as endotoxin. And LPS is the only endotoxin. So if you ever hear endotoxin, think gram-negative bacteria and LPS, because that's it. Now, the other thing that the gram-negatives have that gram-positives don't have is they have pores. And so this becomes important for antibiotics because gram-negative bacteria have an added protection of being able to pump out your antibiotics. So it can help the bacteria be resistant or more resistant naturally to antibiotics. This is not an antibiotic resistant gene. The pore can pump out many things. It can pump in many things, but it is a mechanism they can use. They also have nutrient binding proteins, siderophores for iron binding. Those type of things can be used. Now the bacteria have a peptidoglycan layer, but it's very thin. And it's hard for that gram-negative stain to get in there, bind through the alcohol. So that's why in a gram stain, gram-negative bacteria do not stain with that initial purple dye. They can't hold on to it through the alcohol rinse. So this is just a picture showing the two of them. You have gram-positive versus gram-negative. Keep in mind that some virulence factors, such as flagellum, can be in either gram-positive or gram-negative. A capsule can be in either gram-negative or gram-positive, but not all bacterium will have this. Also keep in mind that a lot of the components on these are going to trigger an innate response. And those pattern recognition receptors. Those PRRs, such as toll-like receptors. And so you have peptidoglycan, which is TLR2 with 6 or 1, and you have flagella, which is TLR5. You have LPS, which is TLR4. And so all of these can trigger an innate cell macrophage to target, eat, ingest the bacterium, and it also causes them to trigger inflammation. All right, so let's talk a little bit about peptidoglycan. Peptidoglycan is very important for the bacterium. And peptidoglycan is going to be tightly regulated, both its production as well as its degradation. And both have to occur for the bacterium to be intact. The peptidoglycan layer on the outside is constantly degraded by the bacteria and reformed so that the structural integrity of the peptidoglycan layer is maintained. And so bacteria have enzymes to do this. Bacteria have lysozymes. Spelled that wrong, sorry. And the lysozymes are going to be important for cleaving, for cross-linking, and it's also going to have enzymes that will degrade the peptidoglycan. But let's just talk about peptidoglycan as a structure just very briefly. One of the important things for our antibiotics is this pentapeptide attachment to an N-acetylmuramic acid. So here we have our N-acetylmuramic acid, and here is our pentapeptide. So here's our 1, 2, 3, 4, 5. That's our pentapeptide. Peptide, of course, is just amino acids linked. So in this case, we have alanine, glutamate, lysine, alanine, alanine. And so what's important is the D-alanine, D-alanine. This is a structure part that vancomycin, which is an antibiotic, can block the cross-linking of D-alanine to D-alanine and hence block the formation of peptidoglycan. And so this is the mechanism of action of our vancomycin. Now, peptidoglycan, again, remember it's on both gram-positive and gram-negative. Its formation is going to be the same. You're going to have inside of the cell soluble substrates that are going to be formed. So again, you have that N-muramic acid, which has that pentapeptides, the D-alanine, D-alanine at the end. And then these are going to be building blocks for the peptidoglycan. The peptidoglycan is actually on the outside here, which here's our pentapeptides, our amino acids 5. And then these get cross-linked. So you're going to see that you're going to have cross-linked of different peptidoglycan structures on the outside of the bacterium. So here's the outside. Now, the glycine muramic acid and the muramic acid. So the N-acetylglucosamine and acetylmuramic acid is linked to what is called the bactoprenol. Which is basically going to be what's driving reactions of the transglycosylases. Now, one of the things to keep in mind is that the peptidoglycan is a lot of steps, and so the bacteria wants to recycle as much as possible. So there is a pyrobactoprenol, and this converts the phosphobactoprenol and recycles the backbone. And so you get recycling. And bacitracin blocks recycling. So it blocks that reaction with the pyrophosphobactoprenol. So we have here a few antibiotics that work. We have vancomycin that is going to block the D-alanine cross-linking. We have bacitracin that's going to block the recycling. Oops. And then we're going to, of course, introduce now the penicillin binding proteins that are the target for the beta-lactams. So again, cross-linking is going to be necessary for membrane integrity of your actual bacterium. The enzymes that do this are D-alanine transpeptidases, they're also referred to as carboxypeptidases, and keep in mind that this is in the bacterium for cross-linking. And what these are really called are penicillin binding proteins, or PBPs. Now, when penicillin was first discovered, of course, the mechanism of action was not known. And so it became clear that it was targeting an enzyme, and so these enzymes were just simply called penicillin binding proteins. But now we know they are actually the enzymes that are doing the cross-linking of the peptidoglycan on the exterior side of the bacterium. And so the penicillin binding proteins are the target of penicillin. So penicillin and penicillin-like antibiotics prevent the cross-linking, and so the integrity of the cell membrane is lost. And so all the beta-lactam antibiotics are going to target penicillin-binding proteins. Okay, so moving on to gram-positive only. Gram-positives have teichoic acid and lipoteichoic acid. This is going to be toll-like receptor 2 can target. And so teichoic acid has the ability to provide flexibility. It is going to attract compounds such as calcium and potassium and allow the bacteria to have flexibility. So again, this is TLR2. LPS. So LPS is an endotoxin. It, again, is the only endotoxin, so it's only associated with gram-negative bacterium, never gram-positive bacterium. It has 3 structures. It has a lipid A, it has a core polysaccharide, and it has what's called the O antigen. The O antigen is essentially not essential for the bacterium to survive. The other 2 are essential for the bacterium, so it is part of the actual bacterium cell wall. Now, the A antigen is the endotoxin portion, whereas the core is basically what it says, it's a core. And so the O antigen is what differentiates all the different types of E. coli, for example. So E. coli O157 has the number 157 identified O antigens. Neisseria is going to shed LPS. It's going to shed that O antigen, and Neisseria shedding is constant, and so it makes it highly susceptible to complement. And so this is going to be TLR4. People with complement deficiencies are highly susceptible to Neisseria. So they get recurrent Neisseria infections. And that's because complement is so important in punching holes in the bacteria cell that if you lack complement, the bacterium is able to divide. We also have acid-fast bacteria, and this is our mycobacterium. And of course, TB is the famous Mycobacterium tuberculosis. And so basically you have mycolic acid, which is a thick layer, as well as a capsule associated with these bacterium. And so acid fast is basically going to be you are doing a particular stain, the acid fast stain, in which you have first a dye, and this is a carbyl fusion dye. And then you are going to wash with an acid or add an acid. And then you're going to wash, and you do a counter stain of malachite green. And so because of the mycolic acid and the capsule, your mycobacterium is able to maintain that carbyl fusion dye. The acid does not wash it out, so that's why it's called acid fast. Even if you give an acid, it stains, it holds fast to the stain. And then they're going to look this nice little fuchsia pink color. All right, now bacteria are also going to have virulence factors if they're going to cause disease. And typically they're going to have more than one virulence factor. So some of virulence factors are going to be associated with adherence. And so E. coli, streps, and staph are famous normal flora causers of many different infections. E. coli is a gram-negative. Maybe do them in red. E. coli is gram-negative. And then we have, of course, our staph and strep, which are gram-positive. They all have their own ways of adhering to our cells. So E. coli will use Fimbria. Strep has an M protein, staph has an A protein, and they're all going to be important for adhering. They have other functions as well. The M protein is going to protect strep, for example, but one of their main functions is to adhere. Remember, bacteria have to get in and stay in. Some are going to have capsules, true capsules, and it makes them hard to phagocytize. Having that mycolic acid as well in your mycobacterium species make it harder to phagocytize. It doesn't mean that your immune system will never phagocytize them. It just takes it a little bit longer, so the bacteria have more time to replicate and things like that. They're also going to have different enzymes. We have catalase, which of course we use in our peroxisomes to neutralize hydrogen peroxide. And so bacterium also have this particular type of enzyme to neutralize oxygen species. Coagulase is going to help to convert fibrinogen to fibrin to help wall off the immune system so that your bacteria can create a nice little niche for them to grow in. Hydrolases are going to be important for the bacterial cell wall integrity. Hydrolases are going to help the bacteria break down peptidoglycan so that it can be regenerated in order to maintain the integrity of the cell wall. Hemolysin is an exotoxin, so this is not something that is helpful to the bacterium. It's something the bacterium uses in order to break down our cells. And so Clostridium, for example, the Clostridium species, Clostridium tetani, Clostridium botulinum, etc., have hemolysins, which are going to lyse red blood cells, for example. All right, so let's talk about the growth cycle. So I'm going to number the growth cycle just to help discuss the different phases. So we have the first phase, which is called the lag phase, and this is really getting in. So anytime you have a lag phase, this means that you can't really detect it if you were to look in a person's blood or in their tissues because the bacteria is trying to establish themselves. And then you're going to have a period where you're going to have expansion. So this is their exponential phase. And so in this exponential phase, they are taking up nutrients. They are going to be decreasing virulence, which I'll talk about in just a moment. And they are going to be increasing metabolism. They are just happy as clams. They are living the high life, and they are replicating. Now, the exponential phase is where they're most susceptible to antibiotics. And then now you're going to get a plateau phase or a stationary phase where number of bacteria equal resources. And then you're going to get the death phase or the decline phase. And this is where you have a decrease of metabolism and you have an increase of resistance to antibiotics. And this is just due to metabolism. I'm not talking about antibiotic resistant genes. And here you'll also have an increase in virulence factors. And then, of course, this is going to repeat. So there's always a question, why do I have to take these antibiotics for 1 week, 2 weeks, or however long it is? And it's because of this cycle. You're trying to break this cycle, but it takes several rounds of this cycle in order to get rid of all the bacterium. So in this first phase, which is this lag phase, the bacteria consider themselves individuals. They are trying to survive. They're trying to find a place to live. And so they are going to have a high level of virulence. Now, at that second stage where you're getting exponential, this is what we have called quorum sensing, where bacteria are happy, they are sensing other bacteria around them, and now they are becoming part of a quorum, part of a colony. And this is also referred to as a biofilm. And so here they are going to decrease their virulence factors. They don't need them anymore. They're part of this group. And they're going to increase metabolism and work for the colony. And then 3, where you have this plateau, is you have a mix of individuals plus colony. So some individuals are starting to sense there's not enough stuff around here, I need to get more virulent. And others are still happy as clams because they have enough nutrients. And then at the 4th phase, where they're declining, this is where you have a decrease of metabolism and you have an increase of virulence. Again, the bacteria are trying to get in, stay in. They go back to that whole mentality of we are individuals and we're trying to get in. Now, with respect to quorum sensing, keep in mind that bacteria divide by division. They produce daughter cells. It's all going to be linked to their peptidoglycan, so they have to be metabolizing more peptidoglycan in order to grow. And that, of course, all takes energy. So with respect to quorum sensing for extracellular bacteria, you have peptides, so bacteria peptides. And these are going to be a quorum signal. So basically what happens is that you're going to have a small number of bacteria initially. And this is going to, of course, grow to a large number. And then at some point, here's a threshold for quorum where you have enough quorum peptide because you have enough bacterium. So all the bacterium producing this quorum peptide, and it doesn't accumulate to a large enough amount for the bacterium to sense there is a quorum now until there is enough of that quorum signal. Now what the quorum signal does is this is going to alter transcription. So all those virulence factors that it was making, it will turn off. So it turns off virulence and it's going to turn on metabolism. Okay, and so again, the quorum sensing is something intrinsic to the bacterium. So you have, for example, a single strep and it's making its own quorum peptide. And then it has 2 straps and 3, then 4, and now it has 1,000 straps. And now there's enough quorum peptide made by 1,000 straps to signal, hey guys, we have enough around here. Let's start switching our mission here. We're no longer virulent. Now we have a nice little colony, so we want to maintain our colony. And so this is all something internal to these bacterium with these quorum peptides. All right, so really quickly on respiration with respect to bacteria. So cellular respiration, of course, is going to be important in generating ATP. You have aerobic respiration, which is going to use oxygen, and anaerobic respiration that does not use oxygen. And so most of our pathogens are going to be facultative anaerobes. So that means that they can either use oxygen or not use oxygen. Staph is a prime example. It lives on our skin, lots of oxygen around. It gets inside, creates a boil, no oxygen. It's able to divide just as nicely inside that boil as it did out on our skin. Another process is fermentation. Fermentation is defined as the enzymatic breakdown of carbs, in which the final electron acceptor is not oxygen, but another organic molecule. So all bacteria can do glycolysis, whether anaerobic or aerobic. Glycolysis is the mechanism of taking a sugar and forming a molecule called pyruvate. Typically, your sugars are 6 carbon, and pyruvate is a 3 carbon. And then pyruvate can go on to oxidative phosphorylation. And this would be at the cell membrane of the bacteria. In us, it would be in the mitochondria, of course. Or this 3 carbon pyruvate can be used to generate some other fermented product. Some have lactate, and you can make cheese with that. Some are going to have ethanol, and you can make wine. You can have propanoic acid, acetic acid, and this leads to gas gangrene. So this is a fermentation process by Clostridium. You can also have just a release of isopropanol, acetone, carbon dioxide through butyric fermentation. Mixed fermentation can give you different products, acetic acid, succinic acid, etc. Butanol fermentation can give you carbon dioxide and butyl. So all of these processes of fermentation is just basically where you start with your carbs making ATP, and your final electron acceptor, oxygen is not available, so you have to make something else. And these are the different things that bacteria can do with that. And then just a little note about iron metabolism. Iron is needed by all bacteria, and so when they get into a niche, such as our bodies, they are actually going to produce iron binding proteins called siderophores. And E. coli enterobactin is an example of a siderophore. E. coli will actually release this enterobactin. It will go into our bodies, bind up iron, and return back to the E. coli and deliver that iron. And so that's just very important for bacteria metabolism.